Scientists discovered something remarkable in 2025. Stress can spread through social channels and hijack your brain's survival circuits in ways we never understood. When you're around someone who's highly stressed, their anxiety transfers to you through nonverbal cues like facial expressions, body language, and vocal tone. But here's what's even more shocking. This social stress contagion doesn't just make you feel anxious, it actually rewires your brain's neural pathways and forces your body to store fat around your belly as a survival mechanism. The latest neuroscience research reveals that stress contagion is far more dangerous than we ever imagined, affecting everything from your memory to your relationships. What researchers found next changed everything we thought we knew about how stress moves between people. The shocking discovery about stress transfer. The breakthrough came from a research team at Stanford's Neuroscience Institute when they accidentally discovered something that contradicted decades of scientific understanding. Dr. Maria Chen was studying physiological responses in marathon runners when her monitoring equipment detected something unexpected. Observers watching stressed participants were showing identical stress hormone spikes without any direct physical contact. What started as an equipment anomaly turned into the most significant discovery in stress research history. These researchers found that stress doesn't just stay contained within one person, it spreads through social channels and triggers measurable biological responses in everyone nearby. The mechanism works through empathy-driven stress responses that activate your body's stress systems when you perceive distress in others. When someone around you experiences high stress, your brain automatically processes their nonverbal cues, facial expressions, body language, vocal tone, and breathing patterns. This social stress detection system triggers your hypothalamic-pituitary-adrenal axis, causing your own cortisol levels to rise even though you're not directly experiencing the stressful situation. The researchers discovered that this empathetic stress response happens faster and more intensely than previously understood. The timeline shocked even the researchers who discovered it. When they tested volunteers who observed stressed individuals in controlled laboratory settings, cortisol levels in the observers' blood began rising at the 4-minute mark. By the 7-minute mark, these levels had spiked dramatically, sometimes matching or even exceeding the cortisol levels of the originally stressed person. This isn't a gradual process that takes hours or days. Your body absorbs stress from other people faster than most conscious emotional responses occur. What makes this discovery particularly unsettling is that participants had no idea it was happening. The people who developed elevated stress hormones through social exposure reported feeling normal during the initial observation period. They weren't experiencing any obvious symptoms or emotional changes, yet their blood chemistry was being altered at the molecular level. This biological transfer happens completely below the threshold of conscious awareness, which means you could be walking around with elevated stress hormones without knowing where they came from. The 2025 studies tested this phenomenon across multiple demographics and environments. Researchers collected blood samples from 2,847 participants before and after various types of social interactions. The results were consistent across age groups, genders, and cultural backgrounds. In controlled laboratory settings, 73% of people who observed stressed individuals for brief periods tested positive for elevated cortisol markers within 10 minutes. Heart rate monitoring showed synchronized stress responses between stressed individuals and their observers, confirming the biological reality of social stress contagion. These findings completely overturn our understanding of stress as an individual experience. For decades, scientists believed that stress was primarily psychological, something that happened inside your own mind and body based on your personal circumstances and reactions. The discovery of empathy-driven stress transfer reveals that stress operates more like a social phenomenon that spreads through groups automatically. The evolutionary perspective explains why this system exists in the first place. Our ancestors lived in small tribal groups where individual survival depended on group survival. If one member of the tribe encountered a predator or threat, it was crucial for that stress signal to spread rapidly to other tribe members. The person who spotted the danger might not have time to verbally warn everyone, but their stress responses could automatically alert others through visual and auditory cues. This biological early warning system helped entire groups respond to threats simultaneously. But evolution designed this mechanism for occasional life-or-death situations, not for the chronic stress environment of modern life. Today, we're constantly exposed to people dealing with work pressure, relationship problems, financial worries, and health concerns. Every social interaction becomes a potential transfer point for stress responses. Your body's ancient survival system now works against you, constantly flooding your bloodstream with stress signals that have nothing to do with your actual circumstances. The contamination extends beyond direct observation. Researchers found that stress responses can spread through entire office buildings, schools, and public spaces as people unconsciously mirror the stress signals they detect in others. Elevators, meeting rooms, and crowded spaces create environments where stress responses amplify and spread through entire populations. The cumulative effect means you might absorb stress from multiple sources throughout a single day. Temperature and social density affect the transfer rate significantly. Crowded, warm environments increase the intensity of nonverbal stress cues, making social stress contagion more likely. This explains why stress seems more infectious in packed elevators, busy restaurants, or summer outdoor events where people are in close proximity and stress signals are more pronounced. The detection methods developed in 2025 allow researchers to track stress contagion in real time. Advanced monitoring systems can identify stress hormone spikes in groups of people and trace the origin back to the initial stressed individual. These systems reveal how stress spreads through crowds in predictable wave patterns, similar to how epidemiologists track the spread of viruses. Children appear particularly susceptible to social stress contagion. Their developing nervous systems and heightened sensitivity to social cues mean they absorb stress responses faster and in higher concentrations than adults. Classroom environments, family gatherings, and playground interactions become stress transfer points that can significantly impact a child's stress hormone levels throughout the day. Parents dealing with work stress unknowingly trigger stress responses in their children through routine interactions and shared environments. The implications extend far beyond individual health. Social stress contagion represents a measurable, trackable biological phenomenon that connects us all through automatic empathetic responses. Every social interaction becomes an opportunity for biochemical changes that can alter your physiology within minutes. But the most disturbing part isn't just that you absorb stress from others. It's what happens inside your brain the moment those stress hormones start circulating through your system. Your brain's stress response system is hijacked. The moment you witness someone in distress, your brain's empathy circuits activate and trigger your own stress response system through psychological contagion. Your amygdala, the brain's alarm center, doesn't need direct hormone absorption to launch emergency protocols. It responds to the visual and auditory stress signals you detect in others, automatically activating your hypothalamic pituitary adrenal axis as if you were facing the same threat. This empathetic stress response happens through behavioral mimicry and emotional contagion, not biochemical transfer. The neurological pathway operates through mirror neuron systems that evolved to help humans survive in groups. When you observe someone displaying stress signals, rapid breathing, tense facial expressions, elevated vocal tone, or agitated body language, specialized neurons in your brain automatically mirror these patterns. Your amygdala processes these mirrored stress signals as genuine threats, triggering the same cascade of responses it would generate if you were personally experiencing danger. This system helped our ancestors coordinate group responses to predators and environmental threats. Your brain treats empathetically absorbed stress as if it represents a real emergency happening to you right now. The stress signals you detect through observation activate identical neural pathways to those triggered by personal stressors. Brain imaging studies reveal that people watching others in distress show the same amygdala activation patterns as those experiencing their own crises. Your hypothalamus receives these alarm signals and immediately begins flooding your system with stress hormones produced by your own adrenal glands. The cascade effect transforms your entire nervous system within minutes of exposure to others' stress signals. Your HPA axis releases adrenaline, noradrenaline, and cortisol into your bloodstream. Your heart rate increases, blood pressure rises, and breathing becomes shallow. Muscle tension builds throughout your body as it prepares for action. Blood flow redirects from digestive organs toward major muscle groups. Your liver releases stored glucose to fuel immediate responses. Every biological system shifts into survival mode because your brain interprets the observed stress as evidence of present danger. Research from 2025 demonstrates that this empathetic stress activation creates measurable physiological changes in observers. When researchers monitored people watching stressed individuals in controlled settings, they found significant cortisol elevation in observers' blood samples. The stress hormone increases occurred rapidly as participants unconsciously synchronized with the stress patterns they witnessed. Heart rate monitoring showed synchronized stress responses between stressed individuals and their observers, confirming that empathetic stress contagion produces real biological changes. The hijacking process devastates your higher brain functions through the same mechanisms activated by personal stress. When empathetic stress signals activate your amygdala, they simultaneously suppress activity in your prefrontal cortex, the brain region responsible for rational thinking, planning, and emotional regulation. Your decision-making abilities become impaired because your brain prioritizes immediate survival responses over complex analysis. This cognitive suppression happens regardless of whether the stress originated from your own experiences or from observing others. Behavioral mimicry amplifies the stress contagion effect beyond simple observation. Your nervous system automatically begins copying the physiological patterns you detect in stressed individuals around you. Breathing rates synchronize, muscle tension patterns mirror each other, and stress hormone production aligns across groups of people sharing the same environment. This unconscious mimicry creates feedback loops where stress signals bounce between individuals, intensifying the overall stress response in everyone present. Memory formation becomes disrupted when empathetic stress floods your system with the same intensity as personal stressors. Your hippocampus, responsible for encoding new memories, operates less efficiently when cortisol levels spike from observing others' distress. The stress hormones interfere with neural processes that normally transfer information from short-term to long-term storage. You might find yourself forgetting conversations or losing track of tasks after spending time around highly stressed individuals. Emotional regulation becomes nearly impossible when empathetic stress hijacks your brain's normal processing systems. The stress hormones disrupt neurotransmitter balance, causing serotonin levels to drop while stress-related chemicals dominate your neural chemistry. You might experience anxiety, irritability, or emotional overwhelm without understanding the connection to your recent social interactions. Your emotional responses become amplified because your brain operates in the same crisis mode triggered by personal threats. The research reveals that empathetically absorbed stress often produces more intense responses than self-generated stress. Scientists believe this happens because observed stress catches your system unprepared. When dealing with your own problems, your brain has context and can potentially modulate its response. But stress signals from others arrive without warning or explanation, triggering maximum intensity emergency responses that can overwhelm your normal coping mechanisms. Decision-making abilities suffer immediate impairment when empathetic stress activates your survival systems. The suppressed prefrontal cortex cannot perform normal executive functions like weighing options, considering consequences, or inhibiting impulsive reactions. You become more likely to make reactive choices based on emotion rather than logic. Complex problem-solving becomes difficult because your brain redirects resources toward immediate survival functions rather than analytical thinking. Your autonomic nervous system responds to empathetic stress exactly as it would to personal threats. Your sympathetic nervous system dominates while your parasympathetic system shuts down, affecting everything from digestion to creative thinking. This shift channels available energy toward immediate survival functions, leaving fewer resources for higher-order cognitive processes that require calm, focused attention. Most people remain unaware that empathetic stress contagion is affecting their physiology and cognitive function. They might notice feeling more anxious after social interactions, but attribute these changes to emotional factors rather than biological stress responses. The empathetic activation doesn't announce itself with obvious symptoms. It quietly floods your system with the same stress hormones and neural activation patterns triggered by genuine personal crises. The timing makes empathetic stress particularly disruptive to normal functioning. Unlike gradual stress buildup from personal situations, empathetic stress can hit your system suddenly when you encounter distressed individuals. Your brain finds itself flooded with crisis-level stress chemicals without conscious awareness of why this physiological shift is occurring. This creates a disconnect between your actual circumstances and your internal state that can persist long after the social interaction ends. But the neurological hijacking represents only the beginning of how absorbed stress affects your body. The stress hormones circulating through your system after empathetic activation don't just disrupt brain function, they trigger biological processes that can fundamentally alter your metabolism and body composition in ways that most people never connect to their social interactions. The belly fat connection nobody expected. Here's what researchers discovered that completely changes how we think about weight gain. Chronic psychological stress doesn't just mess with your emotions and brain function, it actually triggers your body to start storing fat around your belly through well-documented hormonal pathways. When your own stress response system activates repeatedly from witnessing others' distress, it sends the same survival signals to your metabolism as if you were personally facing ongoing crises. Your body responds by switching into fat storage mode, treating the empathetic stress activation as evidence that danger persists in your environment. The metabolic response happens through your body's own cortisol production triggered by empathetic stress responses. Research shows that chronic stress correlates with 50% higher risk of central obesity because sustained cortisol elevation fundamentally alters how your metabolism processes energy. When you repeatedly witness stress in others, your empathetic responses activate your own HPA axis, flooding your system with the same stress hormones that evolution designed to help you survive emergencies. Your liver starts converting glucose into fat at accelerated rates, while your fat cells become hypersensitive to insulin signals. The evolutionary logic behind this mechanism makes perfect sense when you understand how our ancestors survived. For millions of years, stress meant immediate physical danger: predators, natural disasters, tribal conflicts, or food shortages. When stress hormones appeared in the bloodstream, the body needed to prepare for potential starvation or injury. The fastest way to accomplish this was storing energy as fat around vital organs in the abdomen, creating protective cushioning and energy reserves that could sustain life during extended crises. Your body still operates according to this ancient programming, even when stress originates from empathetic responses to others' problems rather than actual survival threats. The 2025 longitudinal study tracking this phenomenon followed 1,847 participants over 6 months, monitoring body composition changes based on exposure to stressed individuals. The results revealed that people working closely with chronically stressed colleagues, living with stressed family members, or regularly interacting with high-stress individuals showed measurable increases in abdominal fat accumulation. These participants weren't changing their diet or exercise habits, yet their bodies were storing fat as if they were personally experiencing chronic stress through repeated empathetic activation of their own stress response systems. The biochemical process involves complex hormonal cascades that begin when empathetic stress repeatedly activates your own cortisol production. Your stress hormones immediately interfere with insulin sensitivity, making cells less responsive to insulin's normal fat-burning signals. Cortisol activates specific enzymes called lipoprotein lipases, which pull fatty acids from your bloodstream and deposit them into fat cells. Your body also produces more neuropeptide Y, which specifically targets abdominal fat cells and makes them grow larger and multiply faster. Your pancreas responds to elevated cortisol by releasing extra insulin, even when blood sugar levels remain normal. This creates perfect conditions for fat storage because insulin functions as your body's primary storage hormone. When insulin levels spike in cortisol's presence, your metabolism switches completely out of fat-burning mode. Instead of using stored fat for energy, your body starts breaking down muscle tissue while simultaneously storing available calories as new fat deposits. The empathetic stress responses essentially reprogram your metabolism to prioritize survival over maintaining healthy weight. The fat that accumulates from chronic stress becomes particularly stubborn because your body treats it as emergency reserves that shouldn't be accessed unless absolutely necessary. This fat develops different characteristics than normal fat tissue, becoming more densely packed with blood vessels and nerve endings. Your body creates hormonal signals that actively protect this fat from being burned, even during regular exercise or caloric restriction. The stress-induced cortisol essentially tells your metabolism that this fat remains off-limits because it might be needed for survival. Your body prioritizes maintaining and adding to these stress-induced fat stores over burning existing fat from other areas. This creates frustrating situations where people gain weight around their midsection despite maintaining healthy eating and exercise habits. The empathetic stress responses override normal metabolic signals, making your body act as if it's preparing for extended starvation or physical threats. Your metabolism becomes hypervigilant about preserving energy, slowing fat-burning processes while accelerating fat storage mechanisms. Statistical evidence from 2025 research shows that 68% of people with regular contact with chronically stressed individuals develop measurable increases in abdominal fat over 3 months. These increases occurred regardless of participants' age, gender, baseline fitness level, or dietary habits. Researchers controlled for variables like sleep quality, personal stress levels, and physical activity, yet the pattern remained consistent. People experiencing empathetic stress activation were gaining belly fat at rates similar to those experiencing severe personal stress. Workplace implications become staggering when considering how this mechanism operates in office environments. Employees working under stressed managers or alongside chronically anxious colleagues experience repeated empathetic stress activation throughout their workdays. Every interaction with distressed coworkers triggers their own stress response systems, leading to sustained cortisol elevation that promotes abdominal fat storage. Open office designs amplify this effect by increasing opportunities for empathetic stress contagion between coworkers sharing the same environment. Family dynamics create even more intense exposure patterns because of frequent and prolonged interactions with stressed household members. Children experiencing empathetic responses to worried parents, spouses absorbing each other's work stress, and siblings sharing family anxiety all contribute to household-wide metabolic changes. The empathetic stress activation doesn't discriminate based on age or relationship status. It triggers the same fat storage response in everyone who repeatedly witnesses distress in their social environment. The exercise paradox becomes particularly frustrating for people dealing with chronic empathetic stress responses. They might increase workout intensity or duration, expecting to burn accumulating belly fat, only to find minimal results. Chronic cortisol elevation actively interferes with hormonal signals that normally allow exercise to mobilize fat stores. Instead of burning stress-induced belly fat, workouts might trigger additional cortisol production, creating cycles that make weight loss increasingly difficult. The empathetic stress responses essentially transform your body into a survival machine that prioritizes energy storage over everything else, creating stubborn belly fat that resists conventional weight loss methods because your metabolism believes it's protecting you from ongoing environmental threats. But the metabolic changes represent only part of how chronic empathetic stress affects your physiology. The neurological impact goes far deeper than anyone previously understood. Neural pathway rewiring from touch. Your brain's response to chronic stress exposure creates lasting structural changes that fundamentally alter how you think and process information. When you repeatedly witness distress in others, the empathetic activation of your own stress response doesn't just trigger temporary hormonal changes. It begins reshaping your brain's physical architecture through well-documented neuroplastic processes. Research shows that sustained cortisol elevation from any source, including chronic empathetic stress responses, can literally rewire neural pathways and alter brain structure over time. The neurological impact operates through your body's own stress hormone production triggered by repeated empathetic responses to others' distress. When your HPA axis activates repeatedly from witnessing stressed individuals, your elevated cortisol levels begin affecting brain regions responsible for memory, learning, and executive function. Studies demonstrate that chronic stress exposure leads to measurable changes in hippocampal volume and prefrontal cortex connectivity, regardless of whether the stress originates from personal experiences or empathetic responses to others' problems. Your hippocampus becomes particularly vulnerable to damage from sustained cortisol elevation caused by chronic empathetic stress activation. This brain region, crucial for memory formation and spatial navigation, contains high concentrations of cortisol receptors that make it extremely sensitive to stress hormone fluctuations. Research tracking individuals exposed to chronic stress shows hippocampal volume reductions of up to 14% over extended periods, with corresponding declines in memory performance and learning ability. The prefrontal cortex suffers systematic impairment when chronic empathetic stress keeps your cortisol levels elevated for extended periods. This brain region, responsible for executive functions like planning, decision-making, and emotional regulation, becomes less efficient at managing complex cognitive tasks. Neuroimaging studies revealed decreased connectivity between prefrontal regions and other brain areas when people experience sustained stress responses, whether from personal stressors or repeated empathetic activation from social environments. Brain imaging research from major universities demonstrates that chronic stress exposure creates measurable structural changes within months of sustained cortisol elevation. Advanced MRI techniques show reduced gray matter density in areas responsible for memory consolidation and executive function. The white matter connecting different brain regions also shows decreased integrity, making communication between neural networks less efficient. These changes occur regardless of whether stress hormones come from personal experiences or empathetic responses to others' distress. Your amygdala undergoes opposite changes during chronic stress exposure, becoming hyperactive and developing stronger connections to sensory processing areas. While your hippocampus and prefrontal cortex shrink under sustained cortisol pressure, your amygdala grows larger and more reactive. This creates an imbalanced brain architecture where threat detection systems dominate while rational thinking and memory systems become impaired. The empathetic stress responses essentially reshape your brain to prioritize survival functions over higher cognitive abilities. Memory formation networks suffer progressive damage when chronic empathetic stress keeps cortisol levels elevated over time. Your hippocampus normally forms new memories by creating connections between neurons and consolidating information during sleep. But sustained stress hormones interfere with these processes, making it harder to encode new experiences and retrieve existing memories. People exposed to chronically stressed individuals often report feeling mentally foggy because their memory systems are operating under constant chemical interference. The neural pathways responsible for creative thinking and flexible problem-solving become systematically weakened during chronic stress exposure. Your prefrontal cortex maintains connections to temporal and parietal regions that generate innovative solutions and novel insights. Sustained cortisol elevation disrupts these connections, making your brain less capable of thinking outside established patterns. The empathetic stress responses essentially train your brain to rely on automatic, survival-focused responses rather than creative or analytical thinking. Working memory capacity drops significantly when chronic empathetic stress activation keeps your stress response system engaged. This cognitive function, which allows you to hold and manipulate information in your mind, depends on efficient prefrontal cortex operation. Research shows that people experiencing sustained stress perform 25–40% worse on working memory tasks compared to their baseline abilities. The constant low-level activation from empathetic stress responses creates similar impairments by keeping your brain in survival mode. Emotional regulation becomes increasingly difficult as chronic empathetic stress reshapes the connections between your prefrontal cortex and limbic system. The neural pathways that normally help you manage emotional responses become less efficient under sustained cortisol pressure. You might find yourself more reactive to minor frustrations or less capable of maintaining emotional balance during challenging situations. The empathetic stress activation essentially weakens your brain's ability to modulate its own responses. Sleep-dependent memory consolidation suffers when chronic empathetic stress keeps your nervous system in heightened activation states. Your brain normally processes and stores memories during deep sleep phases, but sustained stress responses interfere with these restorative processes. People regularly exposed to stressed individuals often experience fragmented sleep patterns that prevent proper memory consolidation, creating cumulative cognitive deficits over time. The recovery process requires deliberate intervention because your brain adapts to chronic stress by treating the altered state as normal. The structural changes don't automatically reverse when stress exposure decreases. Your brain has literally rebuilt itself around the assumption that heightened vigilance is necessary for survival. Rebuilding healthy neural networks requires consistent effort and often professional intervention to overcome the ingrained patterns created by chronic empathetic stress responses. These neurological changes create cascading effects that extend far beyond simple stress responses, fundamentally altering how your brain processes and stores information on a daily basis. The memory and cognitive destruction. The cognitive destruction begins immediately when chronic stress exposure activates your own stress response system repeatedly through empathetic reactions to others' distress. Your hippocampus, the brain region crucial for memory formation, becomes the primary target of your body's own elevated cortisol production triggered by witnessing stressed individuals in your environment. This brain structure contains high concentrations of cortisol receptors that make it extremely vulnerable to damage from sustained stress hormone exposure, whether that stress originates from personal experiences or empathetic responses to others' problems. When your HPA axis activates repeatedly from observing distressed people around you, the resulting cortisol elevation systematically dismantles the neural machinery responsible for forming and retrieving memories. Your hippocampus contains specialized cells that work together to encode new experiences into long-term storage, but sustained stress hormones interfere with the protein synthesis your brain needs to solidify new memories. The elevated cortisol blocks the delicate chemical processes that normally transfer information from short-term memory into permanent storage, leaving you in a state where information enters your mind but never gets properly consolidated for later retrieval. The mechanism operates by disrupting long-term potentiation, the process your hippocampus uses to strengthen synaptic connections between neurons when forming new memories. This requires specific proteins and neurotransmitters working in precise coordination, but chronic stress hormones trigger the release of enzymes that break down the very proteins your brain needs for memory consolidation. Your hippocampus essentially becomes unable to lock in new information because the sustained cortisol has sabotaged its storage mechanisms through repeated activation from empathetic stress responses. Research conducted throughout 2025 tracked cognitive performance in individuals experiencing chronic stress through various sources, including repeated exposure to distressed individuals in their social environments. The results showed consistent reductions in memory performance across multiple testing scenarios. Participants dealing with sustained stress struggled to remember information, failed to recall details from recent experiences, and showed dramatic drops in their ability to form new memories. These weren't subtle changes requiring sophisticated testing to detect. The memory impairments were severe enough to affect basic daily functioning. The fog-like state that people describe after prolonged exposure to stressed environments has a clear neurological basis rooted in how chronic cortisol affects information processing speed. Your brain normally filters and prioritizes incoming information through your prefrontal cortex before routing it to appropriate storage areas. Sustained stress hormones slow down this filtering process dramatically while simultaneously reducing the efficiency of neural communication throughout your brain. Information gets stuck in processing loops, creating the sensation that your thoughts are moving through thick liquid rather than flowing freely from one idea to the next. Working memory takes perhaps the hardest hit when chronic stress keeps your cortisol levels elevated over extended periods. This cognitive system allows you to hold multiple pieces of information in your mind simultaneously while manipulating and combining them to solve problems or make decisions. Sustained stress hormones disrupt the neural networks that maintain working memory by interfering with the rhythmic firing patterns of neurons in your prefrontal cortex. Your ability to keep track of multiple variables, follow complex conversations, or work through multi-step problems drops dramatically because your brain can no longer maintain the active neural patterns necessary for working memory function. Sleep-based memory consolidation suffers devastating interference from chronic stress because sustained cortisol disrupts the precise sleep architecture necessary for transferring memories from temporary to permanent storage. During normal sleep, your brain cycles through specific stages that each serve different memory functions. Chronic stress hormones prevent your brain from entering the deeper sleep stages where memory consolidation typically occurs. The elevated cortisol keeps your nervous system in a state of hypervigilance that blocks the neural processes responsible for strengthening memory traces and integrating new information with existing knowledge networks. People experiencing chronic stress consistently report feeling mentally cloudy and unfocused because sustained cortisol creates widespread disruption in neural communication networks. The stress hormones don't just affect specific brain regions, they interfere with the global neural oscillations that normally coordinate activity across different brain areas. Your neurons begin firing in chaotic patterns rather than the synchronized rhythms necessary for clear thinking. This creates a subjective experience of mental fog where thoughts feel disconnected, concentration becomes effortful, and mental clarity feels constantly just out of reach. The impact on work performance becomes immediately apparent when chronic stress sabotages the cognitive abilities that professional success depends on. Complex problem-solving requires integrating information from multiple sources, generating creative solutions, and maintaining focus over extended periods. Sustained cortisol undermines each of these capabilities by disrupting the neural networks responsible for executive function. Employees dealing with chronic stress from their work environment find themselves making more mistakes, missing important details, and struggling to maintain productivity levels that were previously effortless. Daily decision-making becomes increasingly difficult as chronic stress systematically impairs the mental processes required for good judgment. Every decision requires evaluating options, predicting consequences, and integrating current circumstances with past experiences. Sustained cortisol disrupts each step of this decision-making process by impairing working memory, blocking access to relevant memories, and reducing the cognitive flexibility necessary for considering multiple perspectives. Simple choices that normally require minimal mental effort become surprisingly challenging when your cognitive systems are operating under chronic stress influence. The cumulative effect creates a vicious cycle where cognitive impairment leads to additional stress, which then makes you more susceptible to empathetic stress responses from others in your environment. Your reduced mental performance makes you more vulnerable to absorbing stress signals from your social surroundings, while simultaneously making it harder to implement effective coping strategies. But understanding how this cognitive destruction unfolds reveals something even more disturbing about how stress moves through the people closest to you. Relationship contamination patterns. Close relationships create the most powerful pathways for stress transmission because they involve frequent emotional synchronization and shared environmental stressors. Your intimate partnerships, family connections, and closest friendships generate the strongest empathetic responses, making you most susceptible to absorbing stress through emotional contagion rather than physical contact. When your romantic partner experiences workplace pressure or personal anxiety, your empathy systems automatically mirror their distress signals, triggering your own stress response and creating synchronized cortisol patterns between you. Research tracking couples over extended periods reveals how intimate partners develop correlated stress hormone rhythms through shared daily routines and emotional experiences. When one partner faces chronic work stress, their elevated anxiety levels create an environment where both individuals experience similar physiological responses. The empathetic partner unconsciously mirrors the stressed partner's breathing patterns, muscle tension, and emotional state, triggering their own HPA axis activation. Within weeks of cohabitation, couples show measurable cortisol synchronization as their empathetic responses align their stress cycles. Family contamination follows predictable patterns based on emotional proximity and caregiving relationships. Parents dealing with financial worries, work pressure, or personal crises unconsciously transmit their stress to children through emotional contagion rather than physical contact. Children's developing empathy systems make them particularly sensitive to parental distress signals, causing them to absorb and mirror their parents' anxiety levels. The empathetic responses trigger stress hormone production in children's own systems, affecting their sleep patterns, academic performance, and emotional regulation without any direct hormone transfer. Workplace stress contagion operates through emotional mimicry and shared environmental pressures that create synchronized stress responses across teams. When managers experience high-pressure situations, their visible anxiety triggers empathetic responses in employees who unconsciously mirror their supervisor's stress signals. Open office environments amplify this transmission because workers continuously observe each other's stress behaviors throughout the day. Team meetings become stress synchronization events, where a single anxious leader can trigger empathetic stress responses in entire groups through emotional contagion. The social network effects extend beyond direct relationships through what researchers call emotional contagion cascades. When you absorb stress through empathetic responses to a colleague's anxiety, your elevated stress levels make you more likely to transmit similar emotional signals to your family members. Your spouse empathetically responds to your work-related stress, then unconsciously displays stress behaviors that trigger responses in your children. This creates measurable stress patterns that ripple through communities as empathetic responses spread through interconnected social networks. Groups of stressed individuals create amplification effects that transform entire environments into high-stress zones through collective emotional contagion. When multiple team members experience personal problems simultaneously, their combined stress behaviors create an atmosphere where everyone's empathy systems remain constantly activated. Conference rooms and collaborative workspaces become saturated with stress signals from multiple sources, triggering empathetic responses that exceed individual contributions because stress behaviors from different people interact and amplify each other's emotional impact. Personality research reveals distinct patterns in how different individuals respond to emotional stress contagion. Highly empathetic people show greater susceptibility to absorbing stress through emotional mimicry because they automatically attune to others' emotional states more intensely. Their natural tendency to emotionally connect with distressed individuals increases their exposure to stress signals that trigger their own physiological responses. Introverted individuals often absorb stress more intensely, but from fewer sources, because they limit their social interactions to close relationships where emotional contagion effects are stronger. Chronic stress carriers tend to be individuals with ongoing anxiety, demanding jobs, or persistent personal crises who maintain elevated stress levels consistently. These people become walking stress transmission sources, affecting everyone they encounter through continuous emotional signals that trigger empathetic responses. They often remain unaware of their impact on others because their elevated stress feels normal to them. Their visible anxiety, tense body language, and stressed vocal patterns continuously activate empathetic stress responses in their social environments. Statistics from couples research demonstrate how quickly intimate partners align their stress patterns through emotional synchronization. Within the first month of cohabitation, partners show measurable cortisol correlation as their empathetic responses create regular opportunities for stress synchronization. By 3 months, 85% of couples tested displayed synchronized cortisol patterns that rose and fell together throughout daily cycles. The synchronization becomes so complete that partners often experience identical stress symptoms despite facing different personal challenges. Your social circle essentially determines your baseline stress levels through accumulated daily exposures to others' emotional states. People surrounded by calm, emotionally regulated friends and family members experience fewer empathetic stress responses and maintain more stable personal stress patterns. Conversely, individuals embedded in high-stress social networks absorb elevated stress through continuous empathetic activation, creating chronic physiological stress that persists regardless of their personal circumstances. Professional environments create particularly intense contamination patterns because workplace relationships combine frequent interaction with performance pressure and shared stressful experiences. Stressed executives transmit their anxiety to staff through visible stress behaviors that trigger empathetic responses. Anxious team members spread their stress through collaborative projects that require regular communication and emotional interaction. Customer service roles create exposure to stress from external sources, as employees empathetically respond to frustrated clients' emotional states. The mapping technology developed in 2025 allows researchers to track stress contagion patterns within organizations and communities by monitoring synchronized physiological responses across social networks. These patterns reveal that stress contagion operates through predictable emotional transmission pathways, following social connection routes and creating measurable synchronization effects that can be tracked and potentially interrupted through targeted interventions. But the synchronized stress responses between people create consequences that extend far beyond emotional discomfort, triggering biological changes that systematically compromise your body's most essential defense systems. The immune system collapse. Chronic stress exposure systematically dismantles your immune system through well-documented biological pathways that leave you vulnerable to infections and disease. When your HPA axis activates repeatedly from witnessing distressed individuals in your environment, the resulting elevation in your own cortisol production creates sustained immune suppression that compromises your body's primary defense mechanisms. This isn't temporary discomfort. It's measurable biological damage that accumulates over time as your stress response system remains chronically activated through empathetic responses to others' distress. Your bone marrow responds to sustained cortisol elevation by dramatically reducing the production of new white blood cells, particularly the specialized immune cells responsible for fighting infections and eliminating damaged tissue. The stress hormones trigger cascades that tell your bone marrow to slow down lymphocyte manufacturing while simultaneously increasing neutrophil production immune cells designed for short-term inflammatory responses rather than sophisticated pathogen recognition. This shift leaves you with an immune system poorly equipped to handle complex threats like viral infections or emerging cancer cells. The inflammatory response system suffers immediate suppression when chronic stress keeps your cortisol levels elevated over extended periods. Your immune system normally uses controlled inflammation to isolate threats and coordinate healing responses, but sustained cortisol disrupts this process by blocking molecular signals that activate inflammatory cascades. Your body loses its ability to mount appropriate inflammatory responses to injuries, infections, or tissue damage because the elevated stress hormones have disabled the communication networks that coordinate immune responses. Meta-analyses of stress and immune function research consistently demonstrate that chronic stress exposure significantly impairs immune cell formation and inflammatory responses across diverse populations. Studies tracking thousands of participants over extended periods show that individuals experiencing sustained stress display measurably weakened immune function, with infection rates 60% higher than those with minimal stress exposure. These findings hold true regardless of whether stress originates from personal circumstances or empathetic responses to stressed individuals in social environments. The double immune hit creates particularly devastating scenarios where your immune system faces assault from multiple directions simultaneously. When you're dealing with personal stressors while also experiencing empathetic stress responses from others around you, your immune system experiences compound suppression that exceeds damage from either source alone. Your body's defense mechanisms become overwhelmed by the combined stress load, creating gaps in immune surveillance that leave you vulnerable to opportunistic infections and accelerated disease progression. Your gut microbiome suffers severe disruption when chronic stress alters the delicate bacterial balance that supports immune function. Sustained cortisol changes pH levels in your intestinal tract while simultaneously reducing beneficial bacteria production that normally helps train and support immune responses. These bacterial populations play crucial roles in immune system development and pathogen resistance, so their disruption creates cascading effects throughout your entire defense network. Research documents alarming increases in autoimmune flare-ups among people experiencing chronic stress through various sources. Sustained stress hormones don't just suppress immune function, they can misdirect immune responses toward your body's own healthy tissues. Your immune system becomes dysregulated by chronic cortisol signals and begins attacking joints, organs, and other tissues as if they were threats. People with existing autoimmune conditions show significantly more severe symptoms during periods of chronic stress, while others develop new autoimmune problems they had never experienced before. Cellular aging acceleration becomes visible through measurable changes in telomere length among people chronically exposed to stress. Telomeres are protective DNA sequences that cap chromosome ends and normally shorten gradually with age. Chronic stress hormones speed up the shortening process dramatically, causing cells to age faster than under normal circumstances. Your immune cells are particularly vulnerable to this accelerated aging because they divide frequently and depend on healthy telomeres to maintain protective functions. The connection between chronic stress and cancer risk operates through breakdown of immune surveillance mechanisms that normally identify and eliminate abnormal cells before they develop into tumors. Your immune system constantly patrols your body looking for cells showing signs of cancerous transformation, but sustained cortisol disrupts this surveillance by reducing natural killer cell activity in other specialized immune populations. Cancer cells that would normally be detected and destroyed in early stages can evade immune detection and begin proliferating unchecked. Chronic stress creates immune exhaustion where defense systems become less responsive to genuine threats over time. Constant elevated cortisol forces your immune system to maintain high alert status indefinitely, eventually leading to immune burnout where white blood cells become less efficient at recognizing and responding to actual pathogens. This exhaustion leaves you increasingly vulnerable to infections your immune system would normally handle easily. Sleep-related immune functions suffer severe disruption when chronic stress interferes with natural immune system maintenance that occurs during rest periods. Your body normally uses sleep time to produce new immune cells, clear metabolic waste from immune tissues, and consolidate immune memories that help fight future infections. Sustained cortisol prevents your immune system from completing these essential maintenance functions, leaving you with compromised defenses that become progressively less effective over time. The cardiovascular implications of immune suppression from chronic stress create additional health risks as your compromised immune system struggles to maintain healthy blood vessels and heart tissue. Chronic immune dysfunction allows inflammatory processes to damage your cardiovascular system while simultaneously reducing your body's ability to repair this damage effectively. The combination creates accelerated cardiovascular aging that significantly increases risk of heart disease and stroke. Chronic stress exposure literally transforms your body into a disease-prone environment where infections flourish and healing processes fail, leaving you systematically vulnerable to illnesses that would normally pose minimal threat to a healthy immune system. But understanding these biological mechanisms reveals something crucial about how stress operates and potentially how it might be interrupted. Breaking the contagion chain. The most exciting discovery of 2025 research isn't just understanding stress contagion. It's learning that you can actually interrupt it using evidence-based strategies that strengthen your natural resilience systems. Scientists found that specific stress management techniques create measurable protection against empathetic stress responses by training your nervous system to maintain stability even when surrounded by distressed individuals. These interventions work by strengthening your parasympathetic nervous system and improving your ability to regulate emotional responses automatically. Mindfulness meditation provides one of the most powerful defenses against stress contagion by training your brain to observe others' distress without automatically mirroring their physiological responses. Regular meditation practice strengthens your prefrontal cortex's ability to regulate emotional reactions while reducing amygdala reactivity to stress signals from your environment. Research shows that people with consistent meditation practices absorb significantly less empathetic stress because their brains have developed stronger emotional regulation networks that can maintain stability during social stress exposure. The meditation technique requires daily practice to build protective effects against stress contagion. You focus attention on your breath while observing thoughts and emotions without judgment, including empathetic responses to others' stress. When you notice yourself beginning to mirror someone else's anxiety or tension, you acknowledge the response without engaging with it emotionally. This mental training creates automatic emotional boundaries that prevent empathetic stress from triggering your own stress response systems. Regular aerobic exercise creates biological resistance to stress contagion by optimizing your stress response system's recovery capacity. Physical activity increases production of brain-derived neurotrophic factor, which protects neural tissue from cortisol damage while improving your brain's ability to return to baseline after stress exposure. People who maintain consistent exercise routines show faster cortisol clearance and reduced empathetic stress responses because their bodies have developed more efficient stress processing mechanisms. The exercise protocol needs sufficient intensity to trigger protective neurobiological changes. Moderate to vigorous aerobic activity for 30 to 45 minutes, performed 4 to 5 times weekly, creates optimal stress resistance. During exercise, your body practices rapid stress activation followed by recovery, essentially training your nervous system to handle stress more efficiently. This conditioning makes you less reactive to empathetic stress signals, while improving your ability to return to calm states quickly after social stress exposure. Time in natural environments provides measurable protection against stress contagion through multiple biological pathways that promote nervous system regulation. Nature exposure reduces cortisol production while increasing parasympathetic nervous system activity, creating internal conditions that resist empathetic stress responses. Studies demonstrate that people spending regular time outdoors show reduced stress hormone reactivity and improved emotional regulation when exposed to stressed individuals in their social environments. The nature intervention works most effectively when practiced consistently rather than occasionally. Spending at least 20 minutes in natural settings 3 to 4 times weekly creates cumulative stress resistance that builds over time. During nature exposure, your nervous system automatically shifts toward parasympathetic dominance, which strengthens your baseline resilience against future stress contagion. The combination of fresh air, natural light, and green environments triggers multiple biological systems that support stress recovery and emotional stability. Sleep optimization creates fundamental protection against stress contagion by maintaining healthy cortisol rhythms and supporting emotional regulation systems. Quality sleep allows your brain to process and clear stress hormones while consolidating emotional memories in ways that reduce future stress reactivity. People with consistent sleep schedules and adequate sleep duration show significantly less empathetic stress absorption because their nervous systems maintain better baseline stability throughout daily social interactions. The sleep protocol requires both duration and timing consistency to provide stress protection. 7 to 9 hours of sleep within a regular schedule supports optimal cortisol rhythm regulation while allowing sufficient time for stress hormone clearance during rest periods. Poor sleep disrupts these protective mechanisms, making you more susceptible to absorbing stress from others because your nervous system lacks the resources needed to maintain emotional boundaries during social interactions. Social support networks provide crucial buffering effects against stress contagion by creating environments where positive emotional states can counteract negative empathetic responses. Relationships with emotionally stable, supportive individuals help regulate your stress responses through positive social contagion that works in the opposite direction from stress transmission. When you're surrounded by calm, regulated people, your empathy systems mirror their stability rather than absorbing stress from more anxious individuals in your environment. Building protective social connections requires intentional cultivation of relationships with people who demonstrate consistent emotional regulation and positive coping strategies. These relationships create safe spaces where your nervous system can recover from stress exposure while learning healthier response patterns through positive modeling. The emotional regulation you observe in supportive relationships strengthens your own capacity to maintain stability when encountering stressed individuals in other social contexts. Journaling provides targeted intervention against stress contagion by helping you identify and process empathetic stress responses before they become embedded in your physiology. Writing about your emotional reactions to others' stress creates conscious awareness of empathetic responses while providing structured methods for emotional processing. Research shows that people who journal regularly about stress experiences show reduced cortisol reactivity and improved emotional regulation when exposed to distressed individuals. The journaling technique works by creating psychological distance between your personal emotional state and the empathetic responses triggered by others' stress. You write about the specific situations where you absorbed stress from others, identifying the physical sensations and emotional changes you experienced. This conscious processing helps your brain categorize empathetic responses as separate from your personal stress, reducing the likelihood that future empathetic activation will trigger your own stress response systems. These evidence-based interventions work synergistically when combined into comprehensive stress protection protocols that address multiple aspects of your stress response system simultaneously. Using meditation for emotional regulation while maintaining exercise routines and optimizing sleep creates multiple layers of protection that are significantly more effective than any single intervention alone. The combination approach allows you to prevent empathetic stress absorption while rapidly recovering from exposures that do occur. But even with the strongest protective strategies, some stress damage inevitably accumulates over time through unavoidable social exposures and personal stressors. The remarkable news is that your brain possesses an extraordinary capacity to repair this damage and rebuild itself in ways that researchers are only beginning to understand. The Neuroplasticity Recovery Protocol. Specific neuroplasticity fostering activities can help your brain recover from chronic stress damage by stimulating the growth of new neural connections and strengthening existing pathways. Your brain maintains the ability to reorganize itself throughout your lifespan, creating opportunities to rebuild cognitive functions that stress has impaired. Research shows that targeted interventions can harness this neuroplasticity to restore memory, attention, and emotional regulation, capabilities that chronic stress exposure has compromised. Learning new skills provides one of the most powerful methods for stimulating neuroplasticity and rebuilding stress-damaged brain regions. When you acquire unfamiliar abilities like playing a musical instrument, speaking a foreign language, or mastering complex crafts, your brain creates entirely new neural networks while strengthening connections between different brain areas. Language learning particularly benefits executive function by forcing your prefrontal cortex to manage multiple linguistic systems simultaneously. Studies demonstrate that adults who learn new languages show measurable improvements in cognitive flexibility and working memory within weeks of beginning practice. The cognitive demands of skill acquisition force your brain to build fresh pathways around areas that stress has damaged. Musical instrument practice requires coordination between motor, auditory, and cognitive systems, creating multiple opportunities for neural growth and repair. Even 20 minutes daily of instrument practice produces structural brain changes that can be detected through neuroimaging. These activities work by challenging your brain to integrate information across different regions, promoting the kind of neural connectivity that stress typically disrupts. Aerobic exercise creates unique neuroplasticity benefits that specifically target stress-damaged brain regions through mechanisms no other intervention can replicate. Regular cardiovascular activity increases production of brain-derived neurotrophic factor by approximately 13%, providing essential growth signals that support neural repair and development. This protein acts like fertilizer for your brain, promoting new neuron growth particularly in the hippocampus and prefrontal cortex, exactly the areas most vulnerable to chronic stress damage. The exercise protocol needs sufficient intensity and consistency to trigger meaningful neuroplasticity changes. 30 minutes of moderate intensity aerobic activity, performed 4 to 5 times weekly, creates optimal conditions for neural growth and repair. During exercise, your brain increases blood flow to damaged regions while releasing growth factors that support cellular repair processes. The physical activity also helps clear cortisol from your system through metabolic processes, reducing ongoing stress hormone interference with neural function. Resistance training produces complementary neuroplasticity benefits by increasing levels of insulin-like growth factor, another compound that supports neural repair and growth. The cognitive demands of learning new movement patterns force your motor cortex to create fresh neural pathways, while your prefrontal cortex builds new coordination networks. Complex movement activities like dance or martial arts provide even stronger neuroplasticity stimulation because they require integration between multiple brain systems simultaneously. Cognitive challenging tasks specifically rebuild the executive function networks that chronic stress typically impairs most severely. Activities that push your brain slightly beyond its current comfort zone without becoming overwhelming create optimal conditions for neural growth. Chess, strategic puzzles, and complex problem-solving games force your brain to hold multiple variables in working memory while planning several steps ahead. These activities target the prefrontal cortex networks responsible for flexible thinking and emotional regulation. The key to effective cognitive challenges lies in progressive difficulty that adapts to your improving abilities. Starting with simpler tasks and gradually increasing complexity ensures your brain continues building new connections rather than simply strengthening existing ones. Crossword puzzles, Sudoku, and strategic video games all provide structured cognitive challenges that can be scaled to maintain appropriate difficulty levels as your abilities improve. Sleep optimization accelerates neuroplasticity and brain recovery by providing essential time periods when your brain consolidates new neural connections and clears metabolic waste that interferes with proper neural function. During deep sleep phases, your brain increases production of growth factors that support neural repair while simultaneously clearing toxins that accumulate during waking hours. The glymphatic system becomes dramatically more active during sleep, washing away cellular debris that impedes neural communication. Quality sleep requires both adequate duration and consistent timing to support optimal neuroplasticity. Research suggests that 8 hours and 15 minutes represents the ideal sleep duration for most adults to complete full sleep cycles necessary for neural repair. Your brain produces different growth factors and repair compounds during different sleep phases, so getting complete sleep cycles is crucial for optimal neural healing. Interrupted sleep prevents your brain from completing the repair processes that rebuild damaged neural networks. Sleep timing affects neuroplasticity more than most people realize because your brain follows circadian rhythms that optimize different repair processes at specific times. Maintaining consistent sleep and wake times helps synchronize these natural repair cycles with your daily schedule. Poor sleep quality or irregular sleep patterns disrupt the neuroplasticity processes that could otherwise help your brain recover from chronic stress damage. Creative activities provide additional neuroplasticity stimulation by forcing your brain to make novel connections between previously unrelated concepts and ideas. Artistic expression activates regions throughout your brain simultaneously, creating new pathways between areas that don't normally communicate directly. Writing, painting, music composition, and crafts all provide neuroplasticity stimulation that helps repair the creative thinking networks that chronic stress systematically attacks. The effectiveness of creative activities for neuroplasticity depends on engaging with unfamiliar creative challenges rather than repeating familiar artistic activities. Learning new creative skills forces your brain to build entirely fresh neural networks rather than simply strengthening existing ones. Even simple activities like sketching objects from unusual angles or writing stories in unfamiliar genres provide meaningful neuroplasticity stimulation that supports brain recovery from stress damage. These evidence-based approaches work by harnessing your brain's natural capacity for growth and repair, creating measurable improvements in cognitive function that can counteract the damage caused by chronic stress exposure. But while individual neuroplasticity interventions help repair existing damage, researchers have discovered that the environments where we live and work play an equally crucial role in either promoting or preventing stress-related brain damage in the first place. Creating stress-resistant environments. Simple environmental modifications can create meaningful protection against stress contagion by reducing the psychological and physiological factors that make you more susceptible to absorbing other stress responses. Your physical surroundings influence how your nervous system processes and responds to stress signals from people around you, making environmental design a practical tool for building resilience against empathetic stress activation. Proper ventilation provides measurable benefits for stress reduction by ensuring adequate oxygen flow and removing stagnant air that can contribute to feelings of anxiety and overwhelm. Environmental psychology research demonstrates that fresh air circulation helps maintain optimal cognitive function while reducing the physiological markers associated with stress responses. When spaces feel stuffy or poorly ventilated, your nervous system becomes more reactive to stress signals from others because your brain interprets poor air quality as a potential threat that requires heightened vigilance. Opening windows regularly or ensuring mechanical ventilation systems function properly creates environments where your stress response system operates more efficiently. The increased oxygen availability supports better decision-making and emotional regulation, making you less likely to automatically mirror the stress responses you observe in others. Fresh air also helps clear your mind and maintain the mental clarity needed to recognize when you're beginning to absorb stress from your social environment. Natural lighting significantly impacts your body's stress response systems by supporting healthy circadian rhythms and optimizing neurotransmitter production. Exposure to natural light throughout the day helps regulate cortisol patterns, keeping your stress hormones within normal ranges that make you less reactive to empathetic stress signals. Spaces with adequate natural light create psychological environments that feel more open and less threatening, reducing the hypervigilance that makes stress contagion more likely. When natural light isn't available, full-spectrum lighting can provide similar benefits by supporting your brain's natural regulatory systems. The key lies in avoiding harsh fluorescent lighting that can increase stress responses and make you more susceptible to absorbing anxiety from others in your environment. Warm, diffused lighting creates psychological comfort that helps your nervous system maintain stability even when surrounded by stressed individuals. Decluttering your workspace and living areas removes environmental stressors that compound the effects of social stress contagion. Cluttered environments create low-level stress responses that make your nervous system more reactive to additional stressors, including empathetic responses to others' distress. When your physical environment feels chaotic or overwhelming, your brain operates in a state of heightened alertness that makes you more likely to absorb and amplify stress signals from people around you. Organized spaces support cognitive clarity and emotional regulation by reducing the mental energy required to navigate your environment. This preserved mental capacity can then be directed toward maintaining emotional boundaries and resisting empathetic stress responses. Simple organizational systems that keep essential items easily accessible reduce daily friction and help maintain the calm internal state that provides natural protection against stress contagion. Indoor plants provide psychological comfort and stress reduction through biophilic design principles that connect you with natural elements. Research shows that the presence of plants in indoor environments reduces cortisol levels and promotes parasympathetic nervous system activation, creating internal conditions that resist stress contagion. Plants also improve air quality and create visual focal points that can help redirect attention away from stress signals in your social environment. Specific plant varieties like snake plants, pothos, and peace lilies require minimal maintenance while providing maximum psychological benefits. The act of caring for plants creates mindful moments that interrupt stress responses and promote present-moment awareness. These brief interactions with living systems help reset your nervous system and maintain the emotional regulation needed to avoid absorbing stress from others throughout your day. Temperature control plays a crucial role in stress management because your body's thermoregulation systems interact directly with stress response mechanisms. Environments that are too hot or too cold trigger physiological stress responses that make you more reactive to empathetic stress signals from others. Maintaining comfortable temperatures between 68 and 72 degrees helps your nervous system operate efficiently while preserving energy for emotional regulation rather than temperature adaptation. Humidity levels also affect stress responses, with optimal ranges between 40 and 50% supporting comfortable breathing and reducing the physiological strain that makes stress contagion more likely. Dry air can create subtle respiratory stress that compounds empathetic stress responses, while excessive humidity can feel oppressive and increase overall stress reactivity. Simple humidity monitoring and adjustment create environmental conditions that support natural stress resistance. Creating designated quiet spaces within your home or workplace provides retreat areas where you can recover from stress exposure and reset your nervous system. These spaces don't need to be large or elaborate. Even a comfortable chair in a corner with good lighting and minimal distractions can serve as a stress recovery zone. Having reliable access to calm environments helps you process empathetic stress responses before they become embedded in your physiology. The quiet spaces work most effectively when they're consistently available and free from interruptions. Regular use of these retreat areas trains your nervous system to associate specific locations with calm states, making it easier to activate relaxation responses when needed. Even 5 to 10 minutes in a designated calm space can interrupt stress contagion cycles and restore emotional equilibrium after challenging social interactions. Sound environments significantly impact stress contagion by either amplifying or dampening your nervous system's reactivity to other stress signals. Constant background noise from traffic, machinery, or electronic devices creates chronic low-level stress that makes you more susceptible to absorbing additional stress from social interactions. Controlling sound environments through noise reduction or the introduction of calming audio elements helps maintain the internal quiet needed for emotional regulation. Natural sounds like flowing water, gentle rain, or soft instrumental music can mask jarring environmental noises while promoting parasympathetic nervous system activation. These audio elements don't need to be loud or obvious. Subtle background sounds that promote relaxation help maintain psychological environments that resist stress contagion. The key lies in creating consistent, predictable sound environments that support rather than challenge your nervous system's regulatory capacity. These environmental modifications work together to create spaces that support your natural resilience against stress contagion while maintaining the social connections essential for psychological well-being. But understanding how to protect yourself from stress transmission reveals something far more significant about human nature and our potential for collective transformation. Managing your own stress response becomes the most practical defense against the biological damage we've explored throughout this research. When you understand that witnessing others' distress triggers measurable cortisol elevation in your own system, you gain unprecedented control over your mental and physical health through evidence-based interventions. You need to assess your stress exposure immediately, identify the chronically stressed individuals in your daily environment, and recognize when empathetic responses are activating your own stress systems. Start implementing the protective strategies we covered. Consistent mindfulness practice to strengthen emotional regulation, regular aerobic exercise to optimize stress recovery, quality sleep to support neural repair, and strong social connections with emotionally stable people. This knowledge transforms you from passively absorbing stress through empathetic responses into someone who actively protects their cognitive function, immune system, and metabolic health. Understanding stress contagion gives you the power to manage your biological responses rather than letting others' distress unconsciously trigger your own stress cascade.